Hello friends, have a good day. I am Dr. Sagarika Parida, working as Associate Professor in Botany, Centurion University of Technology and Management, Bhubaneswar. Today I am going to start about microorganisms, introduction to microbial world. What is, first of all, we should know what is microorganisms. Microorganisms, micro means small, organism means living organisms. So, very small organisms which are not seen under the by the naked eyes is known as microorganisms. Generally, the living organisms or small organisms less than 1 millimeter are included under microorganisms, and this is not visible by the naked eyes, and we can able to see under the microscope. And believe that, don't worry, microorganisms are helpful, and before billion of years ago the first living organisms are the microorganisms, and they are more helpful to us. 95% of microorganisms are helpful to us. Apart from that, some microorganisms are also there. They cause some of the diseases like sore throats, chicken pox, then loose motion. These are the different diseases caused by microorganisms. Then come to history. First of all, Antony van Leeuwenhoek first he discovered microorganisms from the feces, from soil, from water under his own developed microscope, which is 200 to 300 times magnifications. And these microorganisms, they are small in size, very small in size, but you remember that in a single full stop 900 microorganisms can be present in a single dot, in a single full stop. After a sentence, you are writing a sentence, after that you make a full stop, and in that full stop about 900 microorganisms can stay or can live. And then come to different microorganisms. They are helpful to us. E. coli, Escherichia coli, which is present in our stomach, they are helpful for digestion of food substances inside the stomach. Then streptomyces, another kind of bacteria, they produce streptomycin, one antibiotic, and this streptomycin, this antibiotic, they will help us to cure from several diseases. Then Bacillus acidophilus, this bacteria helps us for curdling of milk, that means conversion of milk to curd. Then come to another type of bacteria, yeast, another type of microorganism, that is yeast. They make the dough for raising the baking industry, to raise the bread porous. Then come to origin of microorganisms. There are two thoughts. One thought is previous Antony van Leeuwenhoek. They thought that the doctrine of spontaneous generation or abiogenesis, that means microorganisms, they originated from abiotic sources, from nature they are originated, and from germ theory of diseases, that cause postulates, which cause one German scientist, they have discovered this cause postulate, and these two theories are there for origin of microorganisms. In the doctrine of spontaneous generation or abiogenesis, the microorganisms, they originated from the nature, from air automatically, and this is proved wrong. And finally, the germ theory of diseases cause postulate was the most acceptable theory of the microorganisms that cause the diseases. There are 4 steps. According to that, the diseases are caused by microorganisms. The first point is that the diseases that caused by the microorganisms, they may be isolated, they can be isolated from the host. Then apart from that, they can be maintained through pure culture. Then third point is that these microorganisms obtained from the pure culture can be injected, can be inoculated to the host, and then the symptoms of the disease should be recorded and compared with the original host. If it is similar, then it is proved that the same type of microorganism is the causal agent of causing the diseases. Then these are about the origin of microorganisms and germ theory of diseases. Then come to the status of microorganisms, where the microorganisms are present in the living world. In the living world, we know that the kingdom system is the highest taxonomical group of all the living organisms, and in that there are different kingdom systems. First of all, Antony van Leeuwenhoek, Carolus Linnaeus, in 1735 proposed two kingdom systems of classification. So, all the living organisms according to Carolus Linnaeus, all the living organisms are classified into two kingdoms. These are Carolus Linnaeus 1735, they classified the living organisms into two kingdoms. These are kingdom Plantae and kingdom Animalia. In kingdom Plantae, all the plants are together grouped into kingdom Plantae and all the animals are grouped into kingdom Animalia. Then three kingdom systems of classification came into existence, and Ernst Haeckel three kingdom system of classification was proposed by Ernst Haeckel in 1866. According to him, all the living organisms are classified into three kingdoms, and these three kingdoms are Protista, then Plantae, kingdom Plantae and kingdom Animalia. In Protista, all the unicellular prokaryotic as well as eukaryotic organisms are placed together, and then in Plantae all the plants, all the herbs, trees are put together, and in Animalia all the animals are put together. So, where is the status of microorganisms? Here in three kingdom systems of classification, the microorganisms, majority of the microorganisms, they are put together in kingdom Protista, whereas still then some of the microorganisms are also included in kingdom Plantae and in kingdom Animalia. The lower forms of plants, the lower forms of animals, they are also present in kingdom Plantae and kingdom Animalia. After that, four kingdom systems of classification came into existence. RF So, all the living organisms, according to Copeland in 1956, he suggested four different kingdom systems, and in that all the living organisms are classified into four kingdoms. First one is Monera, then kingdom Protista, kingdom Plantae and kingdom Animalia. In this case, what Copeland did, he grouped all the unicellular or prokaryotic microorganisms, both eukaryotic and prokaryotic microorganisms are put together in the kingdom Monera. And Monera, this kingdom, they included bacteria and cyanobacteria or blue green algae, and this is also called as BGA. In Monera, bacteria and cyanobacteria are included. They are prokaryotic organisms. You should know what is prokaryotic organism. Pro means primitive and karyon means nucleus. Prokaryotic, these are prokaryotic organisms. Pro means primitive and karyon means nucleus. So, the organisms, they have primitive type of nucleus, that means there is no nuclear membrane, no nucleolus, no nucleoplasm. That means the DNA or the genetic material is present freely in the cytoplasm. There is no membrane bound organelle. These organisms are known as prokaryotic organism, and these bacteria and cyanobacteria comes under that prokaryotic organisms. So, here the living organisms are classified into 4 groups. Then according to him, Protista, they contain or they consist of that eukaryotic unicellular organism. Before coming to eukaryotic, we should know what is eukaryotic. Eu means true, karyon means nucleus. So, eukaryotic organisms, they have true nucleus, that means they have a well defined nucleus in the cell. The cells have well defined nucleus in the center of the cell with the nuclear membrane, nucleoplasm and nucleolus. Within the nucleus, chromosomes or DNA are present. Then and unicellular, the eukaryotic unicellular organisms are grouped into Protista. The examples of Protista are protozoa, dinoflagellates, euglenoids, like that. Then come to the kingdom Plantae. We know that previously also Plantae kingdom is there in binomial system of nomenclature, that is Plantae systems are there in 2 kingdom systems of nomenclature, that is Plantae, kingdom Plantae and kingdom Animalia. So, and all the animals are grouped under Animalia. Then after that 5 kingdom systems of classification was proposed by R.H. Whittaker in 1969. So, according to R.H. Whittaker, all the living organisms are classified into 5 major groups of 5 types of kingdoms. These are Monera, kingdom Protista, and a third group, kingdom Fungi, then kingdom Plantae and kingdom Animalia. Till now 5 kingdoms system of classification is accepted mostly, it is accepted, and in this system another kingdom, the third kingdom is introduced, that is kingdom Fungi, and this kingdom Fungi, the members of the kingdom Fungi, they included different species which lack their eukaryotic, means they possess true nucleus in the cell, but they lack chlorophyll pigments. They cannot photosynthesize. So, they lack chlorophyll pigment or chlorophyll pigments are absent in fungi. Example, a good example is mushroom. We are taking that edible mushroom, we are taking that, and these fungi, they have absorptive mode of nutrition. That means they are either parasitic or saprophytic, mainly they are saprophytic. They degrade the organic decaying molecules, the compounds, into soluble forms, and they take their nutrition by their saprophytic mode of nutrition. That means they inhabit on dead organic matter. And Monera, they are also parasitic, then they may be photosynthetic or chemosynthetic. The monerians, they are photosynthetic or chemosynthetic, means they have chlorophyll pigments, they can synthesize by using light, solar light, and also they can reduce the chemical substances and also synthesize their own food material. And then protista, they have 3 modes of nutrition. On the basis of the nutrition, on the basis of the ecological status, 5 kingdom systems has been made by Copeland and by Whittaker, and according to him, the monerians, they have photosynthetic or they are autotrophic. Autotrophic means they can produce or they can synthesize their own food materials. Then protista, they have 3 kinds of nutritional ability. They may be holozoic, these protista, they have holozoic mode of nutrition, that means they can engulf by pinocytosis. Pinocytosis means the engulf of liquid substances from the surrounding medium and engulf or eating of solid food from the surrounding medium, that is known as holozoic. Then absorptive, they can degrade the organic matter and absorb their nutrients from the outside source. Then ingestion, ingestion means taking of food material or eating something. So, protozoans, they have 3 kinds of nutritional status. Then come to fungi. Fungi, they are eukaryotic, the cells are eukaryotic, true nucleus is present, then they may be unicellular, filamentous, or sometimes they are also mycelial forms. That means network of hyphae, that single mycelia vegetative organ is vegetative part is known as hyphae, mycelial forms. Then these eukaryotic structures, they lack chlorophyll pigments, so that they cannot photosynthesize. Then come to plantae, all the plants are included, and here they are autotrophic, that means they can reduce the carbon dioxide of the atmosphere in presence of chlorophyll pigment, and they can synthesize the food carbohydrates or glucose from carbon dioxide in presence of sunlight, in presence of chlorophyll pigment. And then animals, their mode of nutrition is ingestion followed by digestion. Their mode of nutrition is followed by ingestion or taking of food, and then finally digestion. In kingdom protista, about 2 lakh, in kingdom monera, about 2,50,000 species are present. In protista, 1 lakh species are included under protista. Then in kingdom plantae, 2,50,000 species, and in kingdom Animalia, about 1 million species are present in kingdom Animalia. So, according to, then come to domain system of classification. Before domain system of classification, another system of classification, that is 6 kingdom of system of classification. Woods et al., they proposed in 1977 6 kingdom system of classification, and according to Woods, the monerians, they are divided into 2 subgroups. They are divided into 2 kingdoms, that is kingdom Archaea or Archaeobacteria and kingdom Eubacteria, and rest kingdoms remained same. and this kingdom of system, in this kingdom systems of division, in archaeobacteria the archaebacteria are included, and these archaeobacteria or archaebacteria, they are the most primitive type of bacteria. The archaebacteria, they are the most primitive type of bacteria, and the examples of archaebacteria are methanogens, then halophils, then thermoacidophils, and these methanogens, halophils and thermoacidophils, these methanogens bacteria we found in cows, ruminant of cows it is present, and these bacteria are responsible for conversion of cow dung to methane gas or biogas, which is also called as biogas. And then halophils. What are halophils? Halo means saline or salt. So, halophils are the bacteria those live in, they live in saline soil. And then thermoacidophils. Thermo means temperature. This type of bacteria, they live in high temperature, that is also called as hot springs or Ushna Prasavana, and this bacteria, they can survive more than 100 degrees centigrade of temperature in water, and they are generally found in hot springs. Then come to domain system of classification. After this system of classification, sixth system of classification, then domain system of classification was proposed by, this domain system of classification was proposed by Woos et al. In 1990. According to them, the living organisms are classified into 3 domain systems, that is 3 domains. First one is domain Archaea. Woos et al. In 1990 proposed 3 domain systems of classification. All the living organisms are grouped into or categorized into 3 domains: domain Archaea, domain Bacteria and then domain Eukarya. This domain Archaea, this domain consists of archaebacteria, only archaebacteria. So, domain Archaea consists of archaebacteria, and these archaebacteria, they are the most primitive bacteria, than these bacteria, than the eubacteria, that means true bacteria, they are the most primitive bacteria. And these archaebacteria, there are 3 groups of archaebacteria I have previously discussed, and these are methanogens, then halophils and thermoacidophils. Then third one is thermoacidophils. And methanogens bacteria, they are the main bacteria causing the conversion of methane gas from cow dung, and halophils, these bacteria, they are present in the saline soil or saline environmental condition or saline water. Generally they are present in mangrove vegetation, that is called as Hintalabana, and these bacteria, they can resist high saline condition of soil and water. Nowadays, these halophils and thermoacidophils, these bacteria are taken into consideration for research for developing different hybrid varieties. Then come to thermoacidophiles. These bacteria are present in hot spring. They can withstand or they can live in high temperature, they can live, and these bacteria, they are most primitive than eubacteria or true bacteria, and they are the primitive organism found after the Azoic period. The Azoic period means lifeless. After after origin of the earth, the Azoic period where there was no life, after that these bacteria, archaebacteria, come into existence. Then come to domain bacteria. In this bacteria, all the bacteria, then cyanobacteria and or blue green algae are included in this, both bacteria and cyanobacteria. They are included under domain bacteria. These bacteria, most of the bacteria, the cyanobacteria, some of the cyanobacteria, they have many benefit for human beings. They can be used in medicine industry, medicinal for making medicines. They are also used as nitrogen biofertilizer, because some of the cyanobacteria, they have heterocyst in their cell. Heterocyst is a special kind of cell. Because of presence of heterocyst, this type of bacteria, they can fix the atmospheric nitrogen, and so from the atmospheric nitrogen is fixed into the soil, and the plant can get that nitrogen for its development and growth. And then bacteria also, many harmful bacteria are also there, and most of the bacteria, they are helpful to us for production of antibiotics, for production of different agricultural products, for production of different medicines. These bacteria are helpful, and some of the bacteria, they are also the causal agent or the pathogen for some diseases in plants and animals. Come to domain eukarya. In eukarya, the fungi, plantae, animalia, all the kingdoms are placed in this domain eukarya. Then come to different types of microorganisms. There are basically 5 different types of microorganisms are there. What are these 5 types of microorganisms? These are bacteria, then yeast, then protozoa, then some of the fungi, algae, bacteria, algae, protozoa, fungi and virus. So, these are the different types of microorganisms. Bacteria, you know, algae, some of the algae, they are also known as cyanobacteria or blue green algae. They come into this category. They have chlorophyll pigment, they can photosynthesize, and they are so minute that we cannot see them in naked eyes, and so for that reason they come under algae, that microorganism. Then protozoa, they dinoflagellates comes, Plasmodium falciparum or Plasmodium vivax, that causes the brain malaria, that also come under protozoa. Then fungi, yeast, example is best example for fungi is yeast. Protozoa, dinoflagellates, then Plasmodium, algae, nostoc, then Rivularia. These are the examples. Then bacteria, Bacillus acidophilus is known for making milk into curd, Bacillus. Then Streptomyces, then virus, different virus, poliovirus or polyoma virus. This is the causal agent of polio. Then retrovirus, small herpes virus. So, these are the different category of microorganisms: bacteria, some of the algae, protozoa, then fungi and virus. So, here we can organize different microorganisms into different groups. So, microorganisms, they in every kingdom microorganisms are present and they can be broadly classified into or grouped into kingdom domain Archaebacteria and domain bacteria, and these are different types of microorganisms. Then come to shape of the bacteria. Shape of the bacteria may be different types. It may be spherical, which is called as coccus. Then this spherical or spherical bacteria, rounded bacteria, they are known as coccus. If it is two cells are there, then it is known as diplococcus. And this diplococcus, one example I will give you, Diplococcus pneumoniae is the causal organism of pneumonia. Then if that cocci form is making a linear chain, then it is known as Streptococcus. And this bacteria, if it is present in groups like this, then it is known as Staphylococcus. Staphylococcus. So, these are the bacteria, different types of bacteria according to shape. Then based on the bacteria, they have locomotion also. The locomotion may be gliding or they have flagella. Flagella is meant for locomotion. So, according to the flagella, bacteria are divided into various groups. If before coming to that, the spherical shaped are known as coccus and rod shaped bacteria is called as bacillus. These are rod shaped. If two rods are present, this is bacillus. This is diplobacillus. This is streptobacillus. If rod shaped bacteria forms a chain, then it is known as streptobacillus. So, these are rod shaped bacteria. Apart from that, coma shaped bacteria is there. The form, it forms like a coma. This is known as Vibrio. This coma shaped bacteria just looks like a coma. Then some of the club shaped bacteria are also there. Club shaped bacteria, they have like this with one stalk. So, according to shape of the bacteria are of different types. They may be spherical, which is known as coccus, again divided into coccus, diplococcus, then streptococcus if it is linear chain, then if it is grouped the spherical forms lies in groups, then it is known as Staphylococcus. Like that club shaped, like that rod shaped bacteria are known as Bacillus and Diplobacillus. When two are present in contact with each other, then it is known as Diplococcus. Then if rod shaped bacteria are present linearly, they are present, then it is known as Streptobacillus. Then also some of the bacteria they have coma shaped, and these bacteria are known as Vibrio, and then also some of the bacteria they have club shaped bacteria are there, and these club shaped bacteria looks like this. And apart from this, depending upon the presence of flagella, the bacteria are also of different kinds. Depending upon presence of flagella. Suppose this is the bacterial cell. If at one end flagella is meant for locomotion, if at one end it is there, then it is called as Lophotrichus. Lophotrichus. If the flagella is present both the sides, then this type of bacteria is called as Amphitrichus. That means amphi means both the polar end or both the ends two flagella are present, so it is known as Amphitrichus. Then if the bacterium has flagella all over its body, then it is known as Peritrichus. Peritrichus. Peri means periphery. Throughout the body, entire surface is covered with the flagella. So, it is known as Peritrichus. If there are no flagella is present, then this is known as Atrichus. So, these are different types of bacteria depending upon its size, then depending upon the presence or absence of flagella or the locomotory organ. Then come to algae bacteria. Next type of microorganism is algae. Algae is generally unicellular or multicellular. Unicellular or multicellular. Then they have chlorophyll pigments, they can photosynthesize, and so by this photosynthesis they can synthesize their own food material. They can able to synthesize their own food material. That means they are autotrophic. Auto means self, trophic means nutrition. They can able to produce their own, they can able to synthesize their own nutrition or own food material. Then come to third group, protozoa. They are unicellular organisms. there are near about 40,000 aquatic species and they fed on other living organism. These protozoa they fed on or they depend on other living organisms. Their mode of nutrition, they can take the food by ingestion, then digestion, or some of the protozoa they have absorptive mode of nutrition and some of the protozoa they have holozoic mode of nutrition. Then come to fungi. They may be unicellular or multicellular, then eukaryotic. They have two nucleus, eukaryotic organisms. The chlorophyll pigment is absent. Chlorophyll pigment is absent, so they cannot photosynthesize and therefore their mode of nutrition is heterotrophic. Mode of nutrition is heterotrophic. Hetero means heterotrophic. Hetero means different. So, different mode of nutrition. Most of the fungi they are saprophytic or parasitic and sometimes they are also symbiotic. Parasitic means they depend on living organisms, then saprophytic means they depend on dead organic matter or decaying organic matter and draw their nutrition by decomposing the dead organic matter. Parasitic mode of nutrition means they live on the living host and then draw their food or nutrition from them, and sometimes also they have symbiotic association with some algae. For example, the symbiotic association with some algae, they are lichens. So, these are fungi. Then come to next is virus. Virus is the smallest microbes. So, we have covered bacteria, then algae, then protozoa, then fungi. We have covered bacteria, then algae, fungi, protozoa. Now, we will go to virus, the last microorganisms. Among the 5 types of microorganisms, the last one, the virus. They are the smallest microorganisms. They do not have any cellular organization. They do not have any cellular organization. That means they are subcellular. Means do not have any cellular organization. So, they are considered as the transition between living and non-living organisms. What is transition between non-living? They have some living characteristics and they also have some non-living characteristics. So, the virus they kept in between the living and non-living organisms. So, these viruses, they are living. When they enter into the host cell, they can reproduce. Otherwise, outside the host, they cannot reproduce. But still then, we cannot reduce the status of the virus into chemical substances. The virus particle is made up of only, it consists of only proteins and the genetic material, that is either DNA or RNA, and the protein is the coat of the virus. This is the outer covering or coat. So, virus particle, they do not have any cellular organization, they do not have mitochondria or cell organelles are absent, they do not have nucleus, only protein coat is surrounded to the genetic material. Still the genetic material is either DNA, that is deoxyribonucleic acid, and RNA or ribonucleic acid. So, this is a acid, and also the protein coat is, that is also protein, a chemical substance. Still then we cannot reduce the status of virus into chemicals, because if we take the chemicals like sodium chloride or sugar, it is not multiplicated within the cell. But if the virus genetic material will go to the cell, then it can replicate, that means it can reproduce. So, the virus are kept in the middle or transition zone or transition in between the living and non-living organisms. So, these are different types of microorganisms we have discussed. So, introduction to microbial world. So, you will get some knowledge about what is microorganism, what is its status in the living organisms in the classification, and then different types of microorganisms. So, we will conclude the chapter here. Okay, thank you all.